Welcome back, friends. Welcome again in another video tutorial for microbiology with Shagnik. So in this video tutorial we will going to discuss about the mode of action of the toxin that is secreted by Clostridium tetani. Okay, so in the previous video I have discussed with you about the introduction of the Clostridium tetani. What type of bacteria, what is the properties of the Clostridium tetani have. Okay, so now it is time to see how Clostridium tetani causes diseases in human. Okay, so let me tell you that Clostridium tetani first enter inside the human body in its spore form. That is very first and important thing to know. Okay, and it is entering into the human body through any kind of wound. Okay, just that is known as penetrating trauma, like a puncture wound. And you can simply remember it about that tetanus, our Clostridium tetani, which is in its spore form, can enter inside the body via any kind of puncture or through any kind of puncture or any kind of wound. Okay, so now the question is why they are using the portal of entry, or I should say why they are using the wound as their portal of entry. So the idea is the any kind of puncture wound, especially the deep and very severe wounds, are anaerobic and warm. Okay, and I have told you in the previous video that our Clostridium tetani is obligate anaerobe, right? That means oxygen is toxic to them. That is why those kind of puncture wounds are very favorable for their growth. That is why they uses the wounds as their portal of entry. Okay, so Clostridium spore get introduced from dirty wounds like a rusty nail, as well as clean wound like a freshly washed kitchen knife. Okay, so in fact any kind of wound, okay, that does not mean rusty wound or any kind of clean wounds, that is via clean washed kitchen knife. Along with that, any kind of wound can brings about the risk of tetanus. Okay, and why they are using such kind of wound as their portal of entry? Because those kind of wounds, the environment in those wounds are anaerobic and warm. Okay, that is favorable for the growth of the Clostridium tetani. Okay, now the incubation period, right? Incubation period means after entering the tetanus, after entering the Clostridium tetani, how many time it takes to show its first symptom, right? So it takes near about few weeks to month after the initial injury, right? So that is the first slide about that. In that slide we have discussed that Clostridium tetani entering our body in the form of spore, and they are using puncture wounds such as mainly deep puncture wounds as their portal of entry, because the environment in those wounds are anaerobic and warm, and which is appropriate for their growth, right? And Clostridium tetani requires few weeks to month after the initial injury to show its first symptom, right? So now let's move to the mode of action of the Titanospasmin. Okay, so Clostridium tetani produces a special type of toxin known as Titanospasmin. Okay, it is a kind of exotoxin, and it is again a AB type toxin. Okay, so it have the ability to disrupt the neuromuscular system of the mammals. Okay, so now I want to tell you about two type of paralysis known as flaccid paralysis and spastic paralysis. Okay, so the Titanospasmin is involved in causing spastic paralysis. You can remember it via simple thing that tetanus causes spasm. Okay, that and spasm has similarity with spastic paralysis, right? That is why the spastic paralysis is caused by tetanus, our Clostridium tetani, okay, and the flaccid paralysis is caused by botulinum toxin, okay. So what is the thing? Let me discuss. So this is the neuromuscular system. The actual idea is in the axon terminal. If you can see, you can see various kind of vesicles, okay, and inside the vesicles there are presence of neurotransmitters, okay. So now there are two kind of neurotransmitter. One kind of neurotransmitter are involved in contraction of the muscle, okay, for example acetylcholine, and another kind of neurotransmitter is involved in inhibition of the contraction, okay. So what is the utility of those kind of neurotransmitter which are involved in stopping the contraction? Because remember, once the stimuli, because I think you know that muscle contraction works by whenever there is a small stimuli from the environment, our muscle will contract, right? So after the process of contraction, it is necessary to be relaxed, right? And this relaxation is provided by those kind of inhibitory neurotransmitters, okay, that involved in inhibition of the muscle contraction. So as the contraction is important, along with the inhibition of the contraction is also important, right? So the neurotransmitter which is involved in contraction of the muscle, that is known as acetylcholine, is inhibited, or the release of the acetylcholine is inhibited by the botulinum toxin, that is secreted by Clostridium botulinum, okay. And that means what? That means because I have told you that acetylcholine is involved in the contraction of the muscle, right? So as it prevents the release of the acetylcholine, that means botulism or botulinum toxin is involved in preventing the muscle contraction, right? So the actual idea, let me discuss about the normal thing. So this is the neuron and this is the muscle, okay. So now the idea is, suppose whenever there is a stimuli present in the environment, our vesicles containing neurotransmitter will be activated, okay, and this vesicle will be pulled by a special type of protein called SNARE protein, okay, SNARE, SNARE protein. This SNARE protein will pull those vesicles towards the neuron membrane, and in the neuron membrane, if I zoom this picture, then you can see that there is a special type of protein named syntaxin. The crosstalk between syntaxin and synaptobremin is important for the release of those kind of transmitters, okay, and the crosstalk is facilitated by those kind of SNARE protein, for example SNAP25, okay. And after that crosstalk, neurotransmitters will get the chance to be removed from the neuron, okay, and they will bind with those type of receptors present on the surface of the muscle cell, okay, and then it will in turn facilitate, as you can see there is a acetylcholine receptor, so acetylcholine will bind over here and it will facilitate a series of downstream reaction that will facilitate the contraction of the muscle, okay. And once the muscle contraction is completed, the vesicle containing inhibitory neurons will activated, okay, and they will follow the same mechanism. That means the SNARE protein will pull those vesicles towards the neuron membrane, and there will be a crosstalk between synaptobremin and syntaxin, and that will causes the release of the neurotransmitters. Here the neurotransmitter is inhibitory neurotransmitter, okay, and this inhibitory neurotransmitter will prevent or will inhibit the muscle contraction, okay, and that is how the entire neuromuscular system is going on, okay. The first kind of situation is inhibited by botulinum toxin, which prevents the contraction. As a result, the muscle cannot be contracted even in presence of the stimuli, okay. And the second type of situation, that is the inhibition of the contraction, to prevent excessive muscle contraction is prevented by our tetanus toxin. Okay, so the first kind, the paralysis, which is result by the first type of situation, is called flaccid paralysis. Okay, in which the muscle cannot be contracted even in presence of stimuli, and the second type of situation is known as spastic paralysis, where after stimulation or after contraction due to the stimulation, muscle cannot be relaxed. Okay, so our topic today is to learn about the how the inhibition is going on. Okay, so let's discuss. So first thing is, after entering inside the body, the Titanospasmin toxin is produced by the tetanus, that is Clostridium tetani. Okay, this Clostridium tetani, now this Clostridium tetani after entering inside the host cell, let's say inside the body, it will enter inside a specific type of inhibitory neuron called Renshaw cell. That is very important to remember, that the Titanospasmin will enter inside the Renshaw cell, which is a inhibitory neuron. Okay, that is very important to remember. After entering inside the Renshaw cell, what it will do, it will cleave the Snarry complex. Okay, and I have told you that the Snarry complex is involved in pulling the vesicle containing the neurotransmitter towards the neuron membrane, right? And as Snarry protein is cleaved, can this pulling process will be maintained now? No, and as a result, the release of the inhibitory neuron is prevented. Okay, and as muscle is not getting under such condition a appropriate signal, it will keep firing the signal for muscle contraction. Okay, and as a result, after, despite the absence of stimulus, the muscle continuing the contraction process. Okay, that is the how Titanoplasmin works. Again, I am repeating. So if I repeat the entire process, so the first thing is the Titanospasmin will enter a specific type of inhibitory neuron. Okay, that is named Renshaw cell. Okay, after entering the Renshaw cell, the Snarry protein, which is involved in pulling the in pulling the vesicle containing the neurotransmitter, will be cleaved by the, again I am specifically saying, the A subunit of the AB toxin. Okay, because remember, in the AB toxin, because I have told you that the Titanospasmin is a AB type toxin, among the AB toxin there are two subunits, A subunit and B subunit. B subunit is involved in the binding process. Okay, because Titanospasmin, how Titanospasmin will enter inside the body or inside our inhibitory neuron, there should be a receptor present. This receptor will be interacted with the B subunit. Okay, and as a result, it will be entered via receptor mediated endocytosis, right? So after that, there will be various process by which the A subunit will be separated from B subunit. Okay, and now this A subunit, when it released in the cytoplasm of the inhibitory neuron Renshaw cell, it will cleave the Snarry protein. Okay, which is involved in pulling the vesicle towards the neuron membrane. Okay, and as a result, it will facilitate the spastic paralysis, which causes muscle rigidity and spasm. So that is the overall idea of the how Titanospasmin, which is the toxin produced by Clostridium tetani, do its job to cause spastic paralysis. Okay, so the entire idea, I am one more time repeating, that first of all, it will enter via a puncture of wound in the form of spores, right? Because as it is a obligate anaerobe, And why they are considering the wounds as their portal of entry? Because remember it very very carefully that the environment in the wound is anaerobic and warm, okay, which facilitate the growth of the Clostridium tetani. Once they are inside the body, it will enter the inhibitory Renshaw cell, okay, which is nothing but a inhibitory neuron, okay, and they are entering via receptor mediated endocytosis after the interaction with the B subunit of the AB subunit of Titanospasmin, okay. Once they are inside, the A subunit will be released in the cytoplasm. This A subunit will cleave the SNRI protein, okay, and as a result the SNRI protein can no longer pull the vesicle containing neurotransmitters, okay, and as a result the inhibitory signal of the neuron cannot be transferred to the muscles, okay, and as a result muscle contraction cannot be stopped, which facilitates muscle rigidity and spasm, and this is the process which we known as spastic paralysis, okay. So that is the today's video's topic. So I hope this video will be helpful to you to understand the basic concept of the how Titanospasmin works. So in the next video we will going to discuss about the types of the tetanus. There we will going to see about the generalized tetanus, about the cephalic tetanus, about the neonatal tetanus in very detail, okay. So till then friends, bye bye, take care.